Long before dinosaurs shook the earth with thunderous footsteps, before mammals claimed dominance over continents, and even before reptiles crawled from ancient waters, there existed an age so alien that walking through its landscapes would feel like stepping onto another planet. This was the Carboniferous Period, a time when dragonflies had wingspans wider than hawks, millipedes grew longer than alligators, and scorpions hunted with bodies rivaling those of modern dogs. The air itself was different, supercharged with oxygen levels that would intoxicate any human foolish enough to breathe it. Forests stretched endlessly, not of oak or pine, but of towering scale trees and giant horsetails that created swamps so vast they would eventually compress into the coal seams we mine today. This was the age of insects, an evolutionary experiment lasting 60 million years when arthropods ruled as undisputed masters of land, sea, and sky. The world they inhabited seems impossible by modern standards. A fever dream of gigantism where the laws of biology bent in ways never seen before or since. Imagine a forest floor carpeted with millipedes the length of crocodiles, their segments clicking in rhythmic waves as they consumed rotting vegetation. Picture skies darkened not by birds, but by swarming insects with bodies as large as seagulls, their compound eyes scanning for prey below. Envision shorelines patrolled by scorpions wielding stingers capable of piercing armor. Their claws evolved for combat in an arms race that pushed predator and prey to extremes. What made these creatures grow so impossibly large? How did insects, animals we now crush beneath our shoes, become the dominant life forms on Earth? And what happened to this strange world of giants that seems too bizarre to have ever existed? The answers lie buried in ancient rock formations, preserved in fossilized footprints, and encoded in the chemistry of long-dead atmospheres. The age of insects wasn't just a footnote in Earth's history. It was a biological revolution that tested the absolute limits of what evolution could create. These weren't monsters from mythology or creatures from fiction. They were real animals that crawled, flew, hunted, and died in a world stranger than any fantasy writer could imagine. If you've ever wondered what Earth looked like when insects were the size of monsters and oxygen-fueled giants beyond comprehension, prepare yourself for a journey into the Carboniferous, where evolution created its most extreme experiments and where the impossible became everyday reality. The Oxygen Revolution. The stage for insect gigantism was set by an atmospheric transformation so dramatic that it fundamentally rewrote the rules of biology. During the Carboniferous Period, oxygen levels in Earth's atmosphere soared to concentrations never achieved before or since, reaching approximately 35% compared to today's 21%. This wasn't a gradual shift, but an explosive change driven by the rapid evolution and spread of land plants, particularly the massive scale trees and tree ferns that dominated Carboniferous forests. These ancient plants photosynthesized with relentless efficiency, pumping oxygen into the atmosphere, while simultaneously locking away carbon in their woody tissues. The chemistry behind this oxygen spike reveals why it happened so suddenly. Carboniferous plants evolved lignin, a complex organic polymer that made their trunks rigid and resistant to decay. When these trees died and fell into the swampy environments that covered much of the planet, the bacteria and fungi that normally decompose wood hadn't yet evolved the enzymes to break down lignin effectively. Dead trees simply accumulated in massive layers, their organic carbon permanently removed from the atmosphere and buried in anaerobic swamps. Without decomposition releasing carbon dioxide back into the air, the carbon cycle became severely unbalanced. Photosynthesis continued producing oxygen while decomposition failed to consume it, creating an atmospheric composition unlike anything in Earth's history. This oxygen-rich environment had profound effects on insect physiology. Unlike vertebrates with active circulatory systems that pump blood-carrying oxygen to tissues, insects rely on passive diffusion through a network of tiny tubes called tracheae. Air enters through holes along their body called spiracles, then travels through increasingly narrow tubes until it reaches individual cells. This system works efficiently for small insects, but size creates a problem. The farther oxygen must diffuse, the less efficient the process becomes. In modern atmospheric conditions, this limitation caps insect size at relatively modest dimensions. But in the Carboniferous atmosphere, with oxygen concentrations 65% higher than today, diffusion became dramatically more effective. The physics of gas diffusion follows clear mathematical principles. The rate of oxygen delivery depends on the concentration gradient, the difference between oxygen levels outside and inside the body. Higher atmospheric oxygen creates a steeper gradient, driving oxygen deeper into tissues without requiring any changes to the insect's respiratory system. This meant that tracheal tubes could extend much farther while still delivering adequate oxygen to support cellular metabolism. Insects could grow larger without suffocating from their own size, but oxygen's role extended beyond simple respiration. High oxygen concentrations affected metabolism at the cellular level, providing more fuel for energy-intensive processes like muscle contraction and neural activity. Insect flight, which demands enormous energy output relative to body size, became sustainable even for massive creatures. The hyperoxic atmosphere essentially removed the respiratory ceiling that normally limits arthropod growth, opening evolutionary pathways toward gigantism that would be impossible in today's atmosphere. Fossil evidence confirms this oxygen-size relationship. Researchers have analyzed the dimensions of fossilized insect spiracles, the breathing holes on their exoskeletons, and compared them to body size. In Carboniferous giants, the ratio of spiracle area to body mass was actually lower than in modern insects, meaning their respiratory systems were less developed relative to their size. The only way these creatures could have survived with such proportionally small breathing apparatus was if atmospheric oxygen concentrations were dramatically elevated. The math simply doesn't work otherwise. The timing of oxygen spikes correlates precisely with periods of insect gigantism. Analysis of ancient air bubbles trapped in amber and chemical proxies preserved in rocks shows that oxygen levels peaked during the late Carboniferous and early Permian, exactly when the largest insects appeared in the fossil record. When oxygen levels later declined during the Permian-Triassic transition, insect sizes crashed accordingly. The correlation isn't coincidental, it's causational, driven by the fundamental physics of how insects breathe. This atmospheric revolution also affected insect competitors and predators. Early amphibians and reptiles, which possess more efficient respiratory systems with lungs and circulatory networks, could also benefit from high oxygen levels, but weren't as dependent on them for size increase. However, the hyperoxic atmosphere may have increased fire frequency. More oxygen makes organic matter more flammable. Fossilized charcoal layers from Carboniferous deposits show evidence of frequent wildfires, suggesting that lightning strikes could ignite forests more easily. These fires would have created complex mosaics of burned areas and intact forests, influencing which insects thrived and where. The oxygen revolution wasn't just about enabling giant insects, it was about creating an entire ecosystem operating under different physical rules. Animals metabolized faster, moved more energetically, and existed at sizes that modern atmospheric chemistry simply cannot support. When scientists recreate Carboniferous atmospheric conditions in laboratory chambers and raise modern insects in hyperoxic environments, the insects grow larger than their oxygen normal counterparts, though not to Carboniferous extremes. Evolution requires time to optimize body plans for gigantism, but the principle holds. More oxygen enables bigger arthropods. This transformation represents one of the most extreme examples of how planetary chemistry shapes evolutionary outcomes. The Carboniferous atmosphere wasn't just a backdrop to insect evolution. It was the driving force that made the age of insects possible. Maganura, the dragonfly that ruled the skies. Meganeura remains the most iconic symbol of Carboniferous gigantism, a dragonfly-like predator with a wingspan stretching 75 centimeters, roughly the size of a modern hawk. Its fossils were first discovered in France during coal mining operations, and when paleontologists assembled the fragments, they initially doubted their own reconstructions. Nothing in the modern world prepared them for an insect capable of this scale. Yet multiple specimens confirmed the reality. Meganeura existed, and it dominated Carboniferous skies as the largest flying insect ever to evolve. The wings themselves were engineering marvels. Unlike the membranous wings of modern dragonflies, which are relatively delicate, Meganeura possessed wings reinforced with complex networks of veins that created geometric patterns of stunning intricacy. These veins weren't just structural supports, they contained hemolymph channels that delivered nutrients and allowed the wings to remain flexible yet strong. The wing venation pattern reveals aerodynamic sophistication, with veins positioned to prevent flutter and maintain stability during high-speed flight. Each wing could flex independently, giving Meganeura exceptional maneuverability despite its size. Biomechanical analysis of Meganeura's wing structure shows that it flew differently than modern dragonflies. The wingspan to body mass ratio suggests it was built for endurance rather than explosive acceleration. Modern dragonflies excel at rapid directional changes and hover hunting, but Meganeura likely employed a cruising strategy, soaring over Carboniferous swamps while scanning for prey. Its flight muscles, estimated from attachment scars on fossilized thorax segments, were proportionally massive, capable of sustaining long flights across vast territories. The head of Meganeura was dominated by enormous compound eyes, each containing thousands of individual lenses. These eyes wrapped around the sides of its head, providing nearly 360-degree vision with only a small blind spot directly behind. The visual processing required for flight at high speeds demanded a sophisticated brain, and endocasts of Meganeura skulls show enlarged optic lobes comparable in relative size to modern aerial predators. It could track moving targets while simultaneously navigating through complex three-dimensional spaces. Essential skills for a predator hunting other flying insects. Meganeura's mandibles tell the story of its predatory lifestyle. Unlike modern dragonflies with relatively modest mouthparts, Meganeura possessed powerful jaws lined with sharp, interlocking teeth. These weren't designed for chewing plant matter, but for gripping and dismembering prey. Wear patterns on fossilized mandibles show characteristic scratches from struggling victims, and the jaw muscle attachment points indicate crushing power sufficient to pierce the exoskeletons of other large insects. Meganeura didn't just catch prey, it tore them apart mid-flight. Its hunting strategy likely combined aerial interception with ambush tactics. Carboniferous forests contain numerous flying insects, from primitive mayflies to early beetles and other griffinflies, relatives of Meganeura, that filled various ecological niches. Meganeura probably patrolled flight corridors above swamps and forest clearings, using its superior speed and vision to intercept smaller flying insects. Some paleontologists suggest it may have also perched on tall scale trees, launching surprise attacks on passing prey like modern hawks do from telephone poles. The body plan of Meganeura reveals adaptations for predatory efficiency. Its thorax was densely muscled, housing not just flight muscles, but also the hydraulic systems that powered its legs. When Meganeura caught prey, it formed a basket with its spiny legs, trapping victims against its body, while the mandibles delivered killing bites. Fossilized Meganeura specimens show leg spines worn from repeated captures, evidence of countless successful hunts. Temperature regulation presented unique challenges for an insect this large. Modern insects are ectothermic, relying on environmental heat to maintain body temperature. But Meganeura's flight muscles generated significant metabolic heat during activity, potentially causing overheating problems. The extensive wing venation may have served a dual purpose: structural support and thermal regulation. Hemolymph circulating through wing veins could dissipate excess heat functioning like radiators. During cool periods, Meganeura might have basked like modern dragonflies, spreading its wings to absorb solar radiation. Meganeura's life cycle remains partially mysterious due to limited larval fossils, but evidence from related griffinflies suggests it followed a pattern similar to modern dragonflies. Larvae likely lived in Carboniferous swamps, hunting aquatic invertebrates and small amphibians. The larval stage probably lasted several years, with the immature Meganeura growing through numerous molts, before finally emerging as adults. The final molt would have been spectacular, a creature the size of a crow pulling itself from an aquatic larval skin and expanding wings to full size. The ecological impact of Meganeura cannot be overstated. As apex aerial predators, they shaped the evolution of other flying insects. Prey species developed evasive flight patterns, cryptic coloration, and possibly even primitive mimicry to avoid Meganeura's attention. The presence of such large predators would have created intense selection pressure, driving innovation in insect flight mechanics and defensive strategies. Meganeura's eventual extinction correlates with declining oxygen levels during the Permian period. As atmospheric oxygen dropped, the physiological ceiling for insect size lowered accordingly. Meganeura's descendants became progressively smaller, eventually giving rise to modern dragonflies that retain the same body plan, but at a fraction of the size. The giant griffinflies disappeared entirely, unable to sustain their massive bodies in the changing atmosphere. Meganeura represents not just an evolutionary peak, but an ecological impossibility in modern conditions. A creature that could only exist in the hyperoxic world of the Carboniferous. Arthropleura, the giant millipede. Walking through a Carboniferous forest, the ground itself seemed to move in segments. Arthropleura was a millipede that stretched over 2 meters in length and weighed as much as 50 kilograms, comparable to a modern crocodile, but with dozens of legs rippling in synchronized waves. It remains the largest terrestrial arthropod ever discovered, a creature so massive that its fossilized trackways were initially attributed to small dinosaurs before paleontologists realized dinosaurs hadn't yet evolved. The body of Arthropleura consisted of approximately 30 armored segments, each bearing 2 pairs of legs. These segments were covered in hardened plates made of chitin and calcium compounds, forming a protective shield against predators. The armor wasn't uniform. Dorsal plates were thicker and more rigid, while ventral plates allowed flexibility for the undulating movement millipedes used to walk. Fossilized Arthropleura plates show growth rings similar to tree trunks, allowing researchers to estimate age and growth rates. Some specimens appear to have lived over 15 years, growing continuously throughout their lives. The legs of Arthropleura numbered in the hundreds, though exact counts vary by specimen. Each leg was relatively short but powerful, adapted for pushing the massive body forward through dense vegetation. Unlike centipedes, which are predators with modified front legs forming venomous fangs, millipedes are primarily detritivores, animals that feed on decaying plant material. Arthropleura fit this pattern, though on a scale never seen before or since. Its mouthparts were adapted for chewing tough plant matter, with grinding plates that could process the lignin-rich tissues of Carboniferous plants. Trackways preserved in ancient riverbeds and mudflats provide extraordinary insight into Arthropleura behavior. These trace fossils show the creature moved in straight lines with occasional turns, leaving parallel grooves from its many legs. The spacing between trackways suggests Arthropleura was somewhat social, possibly traveling in loose groups through forest floor environments. Some trackways show resting marks where the animal stopped, curled slightly, and remained stationary, possibly sleeping or digesting food. The diet of Arthropleura has been debated for decades. Fossilized gut contents are rare, but what evidence exists points toward a herbivorous lifestyle. Chemical analysis of coprolites, fossilized feces, attributed to Arthropleura shows high concentrations of plant material, particularly spores, from ancient ferns, and fragments of scaled tree bark. The grinding plates in its mouth weren't designed for meat, but for breaking down cellulose and lignin. However, some researchers argue that Arthropleura might have occasionally scavenged, consuming dead animals when available, similar to how modern millipedes opportunistically feed on carcasses. Respiration in an animal this large presents the same challenges as in giant insects. Oxygen must diffuse through tracheae to reach internal tissues. Arthropleura possessed larger spiracles than modern millipedes, positioned along its body segments. The high Carboniferous oxygen levels made gas exchange efficient enough to support its bulk, but the creature still faced limitations. Arthropleura likely couldn't sustain rapid movement and probably moved at speeds comparable to modern large millipedes, slow but relentless, processing vast quantities of plant material as it cruised through forests. Defense mechanisms in Arthropleura remain partially speculative, but modern millipedes provide clues. Many millipede species secrete toxic or irritating chemicals from glands along their body when threatened. Chemical traces in fossilized Arthropleura segments suggest it may have possessed similar defensive secretions, possibly containing cyanide compounds like some modern relatives. The armor plating provided physical protection against predators, though few Carboniferous predators could challenge a fully grown Arthropleura. Early amphibians and small reptiles might have targeted juveniles, but adults were essentially invulnerable. Molting presented unique challenges for Arthropleura. Like all arthropods, it had to periodically shed its exoskeleton to grow. For an animal weighing 50 kilograms, this process would have been exhausting and dangerous. Fossilized molt remains show that Arthropleura shed its armor in pieces rather than all at once, a strategy that reduced vulnerability. During molting, the animal would have sought sheltered locations away from predators and remained immobile until the new exoskeleton hardened, a process potentially lasting several days. The ecological role of Arthropleura was crucial to Carboniferous forests. As a detritivore, it accelerated the breakdown of dead plant material, recycling nutrients back into the soil. Carboniferous plants accumulated faster than decomposition could process them, leading to massive peat deposits. Arthropleura and other detritivores helped slow this accumulation, though clearly not enough to prevent coal formation. Their feeding activities would have shaped forest floor ecology, creating microhabitats and influencing which plants thrived. Reproduction in Arthropleura likely followed millipede patterns, though no fossils directly confirm this. Modern millipedes engage in elaborate courtship rituals where males transfer sperm packets to females. Eggs would have been laid in soil or rotting wood, hatching into miniature versions of adults. Juveniles probably remained hidden in leaf litter, gradually growing through successive molts until reaching adult size after several years. The disappearance of Arthropleura correlates with environmental changes at the end of the Carboniferous. As oxygen levels declined and climates shifted toward drier conditions, the vast swamp forests that sustained Arthropleura shrank. Without abundant decaying plant material and the hyperoxic atmosphere that enabled gigantism, Arthropleura populations collapsed. Its descendants, modern millipedes, never again approached such colossal sizes, constrained by atmospheric chemistry and ecological competition from vertebrate herbivores. Pulmonoscorpius, the scorpion predator. Evolution has produced many fearsome predators, but few inspire the same visceral reaction as a scorpion the size of a large dog. Pulmonoscorpius kirktonensis achieved exactly that, a terrestrial arachnid stretching 70 centimeters in length, armed with massive pincers and a venomous stinger capable of killing prey far larger than typical scorpion victims. It prowled Carboniferous forests and shorelines as one of the era's dominant terrestrial predators, a creature that demonstrates how oxygen-rich atmospheres enabled arachnids to reach nightmare proportions. The fossil record of Pulmonoscorpius comes primarily from Scotland, where exceptional preservation captured both hard exoskeleton and impressions of soft tissues. These fossils reveal anatomical details that allow paleontologists to reconstruct its biology with unusual precision. The pincers, technically called pedipalps, were powerfully built, with thick segments and robust claw tips designed for crushing rather than delicate manipulation. Muscle attachment scars show that Pulmonoscorpius could generate enormous gripping force, easily capable of cracking the exoskeletons of other arthropods or the bones of small vertebrates. The tail, or metasoma, curved over the scorpion's back in the characteristic scorpion posture. At its tip sat the telson, a bulbous structure containing venom glands and a sharp, curved stinger. Analysis of the telson's internal structure reveals venom ducts large enough to deliver substantial doses of toxin. While the exact composition of Pulmonoscorpius venom cannot be determined from fossils, comparison with modern scorpions suggests it likely contained neurotoxins that paralyzed prey. Given the size of the venom apparatus, a sting from Pulmonoscorpius would have been devastating to animals far larger than insects. Hunting strategies in Pulmonoscorpius likely combined ambush tactics with active pursuit. Scorpions possess sensory organs called pectines, comb-like structures on their underside that detect chemical signals and ground vibrations. Fossilized Pulmonoscorpius show well-developed pectines, suggesting acute sensitivity to prey movements. The scorpion probably hunted by waiting in hiding spots, using its pectines to detect approaching animals, then striking with both pincers and stinger in coordinated attacks. The pincers would grab and immobilize prey, while the tail curved forward to deliver killing stings. The eyes of Pulmonoscorpius reveal interesting adaptations. Scorpions possess both median eyes on top of the head and lateral eyes along the sides. These eyes weren't as sophisticated as those of insects, but they provided adequate vision for nocturnal hunting. Fossilized eye structures in Pulmonoscorpius show it possessed the same arrangement as modern scorpions, suggesting similar behavior patterns, hiding during day, hunting at night. The Carboniferous night would have been darker than modern nights due to the absence of the moon's gradual recession, making scorpion hunting even more dependent on non-visual senses. Respiration in Pulmonoscorpius occurred through book lungs, respiratory organs consisting of stacked plates with blood-filled spaces where gas exchange occurs. Modern scorpions have relatively limited book lung efficiency, which restricts their size in today's atmosphere. But in the Carboniferous hyperoxic environment, book lungs could support much larger bodies. Fossilized book lung structures in Pulmonoscorpius show they were proportionally larger than in modern scorpions, maximized to take advantage of elevated oxygen levels. Diet analysis comes from both direct and indirect evidence. No fossilized stomach contents have been found, but coprolites and feeding traces suggest Pulmonoscorpius consumed a variety of prey. Likely victims included large insects, smaller arthropods, early amphibians, and possibly even juvenile reptiles. The scorpion's size and weaponry made it a versatile predator capable of tackling prey that modern scorpions couldn't threaten. Some researchers suggest Pulmonoscorpius might have scavenged as well, using its powerful pincers to tear apart carcasses. Territorial behavior likely played a significant role in Pulmonoscorpius ecology. Modern scorpions are often aggressive toward each other, and size would have made competition for resources intense. Fossil sites sometimes show multiple Pulmonoscorpius specimens, but whether this indicates social tolerance or simply favorable preservation conditions remains debated. More likely, these scorpions were solitary, defending territories against rivals and coming together only for mating. Mating in scorpions involves elaborate courtship dances where males grasp females with their pincers and guide them across the ground. Pulmonoscorpius probably followed similar patterns, though the size of these animals would have made the process more dramatic. Males would have deposited sperm packets on the ground, maneuvering females into position to pick them up. Females likely gave birth to live young, a trait common in modern scorpions, with miniature scorpions riding on the mother's back for protection during their vulnerable early stages. The exoskeleton of Pulmonoscorpius shows evidence of molting scars and growth patterns. Like all arthropods, it had to periodically shed its armour to grow. Large specimens show numerous molt lines, indicating they lived for several years and grew incrementally. Each molt would have been a dangerous time, with the scorpion vulnerable until its new exoskeleton hardened. Securing safe molting sites away from predators would have been crucial survival behaviour. Defensive capabilities in Pulmonoscorpius were formidable. Few predators would have willingly attacked an animal equipped with crushing pincers and a venomous stinger. Early amphibians might have posed threats to juveniles, but adult Pulmonoscorpius were likely apex predators in their environments. The combination of armour, weapons and aggression made them dangerous adversaries. The extinction of giant scorpions like Pulmonoscorpius followed the same pattern as other Carboniferous giants. Declining oxygen levels made supporting large arachnid bodies increasingly difficult. Additionally, the rise of larger, more sophisticated reptilian predators created competition that giant scorpions couldn't withstand. Modern scorpions never regained the sizes achieved during the Carboniferous, remaining constrained by both atmospheric chemistry and ecological pressures from vertebrate predators. The mayfly swarms. Modern mayflies live brief adult lives, emerging from water to mate and die within hours or days. Their Carboniferous ancestors followed similar patterns, but at scales that transformed entire landscapes. Fossil evidence from ancient lakebeds and river deltas shows mass mortality events where millions of mayflies died simultaneously, their bodies accumulating in layers meters thick. These weren't isolated incidents, but regular occurrences tied to synchronized emergence patterns that created temporary aerial blizzards of insects. Carboniferous mayflies belong to groups long since extinct, though they retained the same basic body plan as modern species. Delicate wings, long tail filaments, and weak flight capabilities. What distinguished them was size and abundance. Some species achieved wingspans of 15 centimetres, making them substantially larger than most modern mayflies. When swarms emerged from Carboniferous lakes and swamps, the sheer biomass involved must have been staggering, temporarily overwhelming local ecosystems with protein-rich insects. The emergent strategy of mayflies represents one of nature's most extreme examples of reproductive synchronization. Mayfly nymphs, the immature aquatic stage, spend months or years developing in water, feeding on algae and organic matter. When environmental triggers align, usually temperature and day-length cues, entire populations molt simultaneously into winged adults. This coordinated emergence serves multiple purposes. It overwhelms predators through sheer numbers, ensures abundant mating opportunities, and concentrates reproductive effort into brief windows of optimal conditions. Fossilized lakebeds preserve the aftermath of these emergencies. Layers of mayfly body fossils show exquisite preservation of wing venation, body segments, and even internal structures. The density of fossils indicates that these mortality layers formed when billions of mayflies died after mating, their bodies falling into water and sinking to oxygen-poor lake bottoms, where decomposition was minimal. Chemical analysis of these layers shows their nearly pure mayfly remains, creating organic horizons distinct from surrounding sediments. The ecological impact of mayfly swarms extended far beyond the insects themselves. Predators from across the food web converged on emergence events. Fish would have gorged on emerging nymphs and fallen adults. Amphibians likely gathered at water's edges to feast on the windfall. Even terrestrial predators like Pulmonoscorpius and early reptiles probably timed their activities to coincide with mayfly emergencies, taking advantage of easy protein sources. The sudden availability of massive food resources would have created temporary feeding frenzies. Birds hadn't yet evolved during the Carboniferous, but aerial predators like Meganeura certainly capitalized on mayfly swarms. The clumsy flight of mayflies made them easy targets, and the sheer abundance meant that even inefficient hunters could catch multiple victims. fossilized mayfly wings showing damage consistent with predator attacks suggest that significant percentages of each emergence cohort died from predation rather than natural senescence after mating. Mating behavior in mayflies is spectacularly brief. Males form aerial swarms above water, flying in coordinated patterns that attract females. When a female enters the swarm, males compete to mate with her, often multiple males attempting copulation simultaneously. The successful male transfers sperm during a brief flight coupling, after which the female immediately seeks suitable egg-laying sites. She deposits eggs on the water surface or on aquatic vegetation, then dies, her entire adult life compressed into hours. The brevity of mayfly adult lives creates intense selection pressure. Adults possess non-functional mouthparts and cannot feed. Their digestive systems are partially filled with air to improve buoyancy during flight. Every physiological system is optimized for reproduction at the expense of longevity. Wings are efficient but fragile, built for single-use flight. Bodies are lightweight, lacking the armor and defenses that protect longer-lived insects. This extreme specialization shows evolution pushed to fascinating extremes, an entire life stage reduced to reproduction and nothing else. Fossilized mayfly nymphs reveal details about their aquatic lives. These immature stages possessed gills along their abdomens, allowing them to extract oxygen from water. Some species show adaptations for burrowing in sediment, while others possess streamlined bodies for swimming. The diversity of nymphal forms indicates that Carboniferous mayflies occupied numerous aquatic niches, from slow-moving swamps to fast-flowing streams. Each environment selected for different survival strategies, creating ecological variety within the broader mayfly lineage. The nymphal stage determined ultimate adult body size. Mayflies that fed successfully and avoided predators grew larger, eventually emerging as bigger adults with better reproductive prospects. Competition among nymphs would have been intense in productive waters, with larger individuals outcompeting smaller ones for food resources. This created size-based hierarchies that persisted from nymphal stages through adult life, influencing which individuals contributed most to the next generation. Mass emergencies also served as nutrient transfer mechanisms. Mayflies spent their nymphal lives concentrating nutrients from aquatic environments, converting algae, bacteria, and organic matter into insect biomass. When adults emerged and died on land, they transported those aquatic nutrients to terrestrial ecosystems. Decomposing mayfly bodies fertilized soils, fed terrestrial scavengers, and enriched environments far from water. This nutrient shuttling linked aquatic and terrestrial food webs in ways that shaped forest floor ecology. The evolutionary success of mayflies during the Carboniferous demonstrates how specialized life strategies can dominate under specific conditions. The vast swamps and lakes of the period provided ideal habitats for aquatic nymphs, while hyperoxic atmospheres supported larger adult bodies. When environmental conditions later changed, oxygen levels declining, wetlands shrinking, mayfly diversity crashed. Modern mayflies retain the same basic life strategy, but at reduced scales, testament to a reproductive pattern refined over 300 million years. Meganeuropsis, the American giant. While Meganeura dominated European skies, North America hosted an even larger predator. Meganeuropsis permiana achieved wingspans approaching 75 centimeters, making it comparable in size to its European cousin, but with subtle anatomical differences that reveal separate evolutionary paths toward gigantism. Fossil discoveries in Oklahoma and Kansas show that giant griffinflies evolved independently on different continents, converging on similar solutions to aerial predation in hyperoxic atmospheres. The wings of Meganeuropsis show distinct venation patterns compared to Meganeura. While both possessed complex networks of supporting veins, Meganeuropsis had slightly different arrangements in the wing base, suggesting differences in flight mechanics. Biomechanical models indicate Meganeuropsis may have been optimized for faster straight-line flight, while Meganeura possessed better maneuverability. These subtle differences likely reflected different prey communities and hunting environments. North American swamps may have favored different predatory strategies than European forests. Fossil preservation of Meganeuropsis rivals that of any Carboniferous insect. Complete specimens show not just wing structures but also body segments, leg positions, and head morphology. The eyes were proportionally larger than in Meganeura, suggesting Meganeuropsis relied even more heavily on vision for hunting. Eye structure analysis reveals thousands of individual lenses arranged in hexagonal patterns, creating compound eyes capable of detecting movement across wide fields of view. This visual acuity would have made Meganeuropsis a devastating hunter of flying prey. The thorax of Meganeuropsis was massively muscled, housing flight muscles that powered its enormous wings. Attachment scars on fossilized thorax segments show muscle arrangements similar to modern dragonflies but scaled up dramatically. Flight in an animal this size required extraordinary power output, and the high oxygen Carboniferous atmosphere provided the metabolic support necessary for sustained aerial activity. Wind tunnel tests using model Meganeuropsis wings show they could achieve speeds comparable to modern hawks, fast enough to intercept most prey and escape most threats. Hunting territories for Meganeuropsis must have been extensive. Modern dragonflies defend territories of hundreds of square meters. An insect the size of Meganeuropsis would have required proportionally larger hunting grounds. The distribution of fossils suggests these predators weren't evenly distributed, but concentrated in areas with abundant prey. River valleys and swamp margins likely served as prime hunting corridors where smaller insects congregated, attracting Meganeuropsis, like modern raptors, gather along migration routes. Prey diversity in North American Carboniferous ecosystems was substantial, providing Meganeuropsis with numerous targets. Primitive mayflies, early beetles, cockroach relatives, and smaller griffinflies all shared the skies. Meganeuropsis likely employed multiple hunting strategies depending on prey type: rapid pursuit for fast-flying insects, ambush attacks from perches for unsuspecting victims, and possibly even low-altitude sweeps over water surfaces to catch emerging aquatic insects. The abdomen of Meganeuropsis was long and segmented, functioning as both a ballast and a rudder during flight. Modern dragonflies use abdominal movements to adjust center of gravity and control pitch and roll. Meganeuropsis would have employed similar tactics, flexing its abdomen to execute tight turns or maintain stability during turbulent flight. The muscular control required for these maneuvers indicates sophisticated neural coordination, with nerve ganglia distributed throughout the body processing sensory information and coordinating movements. Mating in Meganeuropsis probably followed dragonfly patterns, which involve spectacular aerial displays. Males would have established territories, defending them against rival males through aerial combat that combined displays of size and actual physical confrontations. When females entered territories, males would have pursued them in courtship flights, with successful males gripping females behind the head using specialized clasping structures. Mating would have occurred in flight or while perched, with males transferring sperm packets that females stored for later egg fertilization. Egg-laying behavior likely involved females hovering over water surfaces, dipping their abdomens to deposit eggs on aquatic vegetation or directly into water. The larvae, called nymphs, would have been formidable predators in their own right, hunting other aquatic arthropods and possibly even small fish or amphibians. Fossil evidence of dragonfly nymphs from the period shows they possessed extending jaws capable of rapid strikes, a hunting adaptation that modern dragonfly nymphs still employ. The thermoregulation challenges facing Meganeuropsis were more severe than in Meganeura due to its slightly larger size. Maintaining stable body temperature during flight required balancing heat generation from muscle activity against heat loss through the wings. The wing venation system likely served dual purposes: structural support and thermal management, with hemolymph flow adjustable to either conserve or dissipate heat as conditions demanded. Competition between Meganeuropsis and other large predators shaped Carboniferous aerial ecosystems. Multiple species of large griffinflies coexisted, each potentially specializing in different prey sizes or hunting at different times of day. Niche partitioning would have reduced direct competition, allowing several large predators to share the same general environment without driving each other to extinction. This diversity of apex aerial predators has no modern equivalent. Today's largest flying insects are dwarfed by birds, which exclude insects from large predator niches. The geological formations containing Meganeuropsis fossils also preserve evidence of the environments it inhabited. These were coastal swamps and river deltas with complex mosaics of open water, dense vegetation, and occasional dry ground. The climate was warm and humid, with minimal seasonal variation. Such stable conditions supported year-round insect activity, unlike modern temperate regions where winters halt insect reproduction. Meganeuropsis could hunt continuously, maintaining populations without seasonal bottlenecks. The coal forests. The stage upon which Carboniferous giants performed was itself extraordinary, forests unlike anything in the modern world. These weren't woodlands of flowering trees or coniferous evergreens, but alien landscapes dominated by scale trees, giant horsetails, and tree ferns that created canopies up to 40 meters high. The very structure of these forests enabled insect gigantism by producing environments rich in oxygen, dense with vegetation, and largely free from the vertebrate predators that would later constrain insect evolution. Scale trees, technically Lepidodendron and Sigillaria species, formed the backbone of Carboniferous forests. These weren't true trees in the botanical sense, but rather giant club mosses with woody trunks covered in diamond-shaped leaf scars that gave them their scaly appearance. They grew rapidly to heights exceeding 30 meters, producing dense canopies that shaded the forest floor. Unlike modern trees with extensive root systems, scale trees had relatively shallow roots that spread horizontally rather than penetrating deep into soil. This adaptation suited the waterlogged swamps where they thrived, but made them vulnerable to toppling during storms. The reproduction strategy of scale trees was spectacularly wasteful by modern standards. After growing for perhaps 20 years, a scale tree would produce a single massive cone at its crown, sometimes weighing several kilograms and containing millions of spores. After releasing these spores, the entire tree died, having invested all its resources into one reproductive event. This Big Bang reproduction filled Carboniferous forests with standing dead trees, creating complex vertical habitat structures. Dead scale trees provided perches for aerial predators like Meganeuropsis, shelters for ground-dwelling arthropods, and eventually contributed to coal formation when they toppled into swamps. Giant horsetails, calamite species, occupied wetter areas within forests, forming dense thickets along waterways. These plants resembled modern horsetails, but grew to heights of 20 meters, with hollow jointed stems that contained silica deposits, making them abrasive and unpalatable to most herbivores. Horsetail thickets created labyrinthine ground-level environments where smaller arthropods could hide from predators, while the vertical stems provided climbing routes to different forest levels. Tree ferns added another structural layer. Unlike modern ferns that remain relatively small, Carboniferous tree ferns grew substantial trunks that elevated fronds into mid-canopy positions. These ferns reproduced via spores like their modern descendants, creating seasonal dispersal events when mature fronds release clouds of reproductive cells. The spores would have provided food resources for numerous small arthropods, supporting food webs distinct from those feeding on living plant tissue. The forest floor was a dark, humid environment dominated by decomposing plant material. Fallen scale tree trunks, shed leaves, and accumulated detritus created thick organic layers that supported enormous populations of detritivores. Arthropleura thrived in these conditions, processing decaying vegetation alongside countless smaller millipedes, primitive cockroaches, and other decomposer organisms. The sheer volume of dead plant material exceeded decomposition capacity, leading to accumulation of peat deposits that eventually compressed into coal seams still mined today. Light penetration through Carboniferous forests would have been minimal at ground level. The dense canopies blocked most sunlight, creating dim conditions that favoured organisms adapted to low-light hunting and feeding. Scorpions like Pulmonoscorpius benefited from these dark conditions, using chemical and vibrational sensors to locate prey. The forest structure created distinct vertical zones, a sun-drenched canopy, dimly lit mid-levels, and dark forest floors, each supporting different arthropod communities. Oxygen production in these forests was relentless. Photosynthesis occurred throughout daylight hours across millions of square kilometres of forest, pumping oxygen into an atmosphere already enriched by previous centuries of plant growth. The lack of efficient decomposers meant carbon remained locked in wood, rather than returning to the atmosphere as carbon dioxide. This imbalance drove atmospheric oxygen to concentrations that would be toxic to most modern animals, but which supported the gigantic arthropods that define the Carboniferous. Fire ecology played a significant role despite the wet conditions. High oxygen concentrations made organic matter more flammable, and lightning strikes could ignite even damp wood. Fossil charcoal layers appear throughout Carboniferous deposits, showing that fires swept through forests regularly. These fires created patchy mosaics of burned areas, regenerating with fast-growing pioneer species and intact mature forests. Fire-adapted organisms would have colonized burned zones quickly, creating dynamic ecological succession patterns. The absence of large herbivorous vertebrates allowed plants to invest less in chemical defenses compared to modern forests. While some toxins and structural defenses existed, Carboniferous plants didn't face the intense herbivory pressure that shapes modern plant evolution. This meant vegetation was more digestible for arthropod herbivores, supporting larger populations of plant-feeding insects that in turn supported predators like Meganeuropsis. Seasonal patterns in Carboniferous forests remain debated. Much of the Carboniferous world experienced equatorial or near-equatorial conditions with minimal temperature variation, but some regions at higher latitudes may have experienced seasonal changes in rainfall or day length. These variations would have created breeding cycles and migration patterns in arthropods, though nothing comparable to modern temperate insect seasonality. The water cycle in Carboniferous swamp forests was intense. High humidity, frequent rainfall, and limited drainage created perpetually waterlogged conditions. Standing water covered much of the forest floor, providing habitats for aquatic arthropod larvae and amphibians, while simultaneously preserving falling vegetation in anaerobic conditions that prevented decomposition. This hydrological regime was essential for both insect ecology and the coal formation that gives the Carboniferous its name. Eurypterids, the water scorpions. While terrestrial scorpions like Pulmonoscorpius prowled land, their aquatic relatives dominated Carboniferous waters with even more intimidating forms. Eurypterids, commonly called sea scorpions, though many inhabited fresh water, achieve sizes exceeding 2 meters in length, making them the largest arthropods ever to exist in any environment. These predators combine scorpion-like body plans with swimming adaptations, creating animals that ruled aquatic ecosystems as apex predators. The body structure of Eurypterids reveals sophisticated aquatic adaptations. The prosoma, the head and thorax region, contain the brain, sensory organs, and attachment points for 6 pairs of appendages. The first pair formed chelicerae, small mouthparts used to manipulate food. The next 4 pairs served as walking legs when the animal crawled along bottom substrates, while the final pair evolved into broad, paddle-like swimming legs. These swimming legs moved in coordinated strokes that propelled eurypterids through water with surprising speed and agility. The opisthosaur, the abdomen, consisted of 13 segments that tapered to a spike-like telson at the tail. Unlike modern scorpions with venom-delivering tails, most eurypterid telsons lacked venom glands and functioned instead as rudders during swimming and possibly as defensive spines. The abdomen flexed during swimming, adding thrust to the paddle leg strokes and allowing rapid directional changes when pursuing prey or evading threats. Eyes in eurypterids were remarkably advanced. Large compound eyes sat on top of the head, providing excellent vision in aquatic environments where visual hunting offered significant advantages. Some species possessed additional simple eyes, ocelli, that detected light intensity and helped maintain orientation. The combination of compound eyes for detailed vision and ocelli for general light detection gave eurypterids sensory capabilities rivaling modern aquatic predators. The chelicerae and anterior limbs show modifications for prey capture. In some species, the appendages near the mouth possess spines and serrations that could grip struggling prey. The chelicerae worked in coordination with these limbs, tearing food into pieces small enough to consume. Fossilized gut contents from exceptionally preserved specimens show eurypterids fed on fish, other arthropods, and possibly early amphibians. They were opportunistic predators that consumed whatever they could catch and overpower. Size variation among eurypterids was extreme, with some species remaining under 10 centimetres, while giants like Jaekelopterus reached over 2 metres. This size diversity allowed different species to partition prey resources, small eurypterids hunting tiny crustaceans and insect larvae, medium species taking fish and amphibians, and giants capable of attacking anything in their environment. The coexistence of multiple eurypterid species in single water bodies created complex predator hierarchies. Molting in giant eurypterids must have been spectacular events. Shedding an exoskeleton measuring 2 metres required finding secure locations away from predators and competitors. Fossil evidence shows molting eurypterids sought sheltered areas in reefs or under submerged logs. The process would have taken hours, during which the animal was completely vulnerable. Once the new exoskeleton hardened, the eurypterid would have grown noticeably larger, potentially changing its ecological role and relationships with other predators. Reproductive strategies in eurypterids likely involved internal fertilization, with males using modified appendages to transfer sperm packets to females. Some fossilized eurypterids show sexual dimorphism, with males possessing enlarged appendages or body features possibly used in combat or display. Females would have deposited eggs in protected locations, perhaps buried in sediment or attached to aquatic vegetation. Larval eurypterids probably went through numerous molts before reaching adult size, with each stage increasingly predatory. The transition between water and land represents one of eurypterid evolution's most fascinating aspects. While most eurypterids were fully aquatic, some species show adaptations suggesting they could tolerate brief excursions onto land. Strengthened leg joints, modified gill structures for air breathing, and body shapes suited to locomotion on solid surfaces all hint that certain eurypterids experimented with terrestrial lifestyles. These experiments may have influenced later arachnid evolution, with terrestrial scorpions potentially descending from eurypterid ancestors that fully committed to life on land. The extinction of giant eurypterids occurred gradually through the late Paleozoic. Competition from jawed fish with more sophisticated swimming abilities and better sensory systems put pressure on eurypterid populations. The transition from shallow, warm seas to cooler, deeper oceans reduced available habitats. By the end of the Permian, Eurypterids had vanished entirely, their ecological niches filled by fish, marine reptiles, and eventually sharks. Modern horseshoe crabs represent the closest living relatives, though they occupy entirely different ecological roles. Brachyptera, the ancient stoneflies. Among the diversity of Carboniferous insects, stoneflies represent one of the oldest lineages of winged insects, with fossils showing they had already achieved their basic body plan by the early Carboniferous. Brachyptera and related genera demonstrate that even amid the giants, smaller insects continued evolving and specializing. These primitive stoneflies differed from modern species in subtle but significant ways that reveal how insect flight and aquatic life cycles were refined over millions of years. Stonefly fossils preserve wing venation patterns in exquisite detail. The wings of Carboniferous stoneflies show more extensive cross veins than modern species, creating complex geometric patterns that provided structural strength but potentially reduced flight efficiency. Unlike the streamlined wings of modern stoneflies optimized for efficient flight, Carboniferous species possess broader wings better suited for gliding than sustained flapping flight. This suggests they relied more on passive flight between perches rather than active aerial pursuit of prey or mates. The aquatic larvae of ancient stoneflies occupied similar ecological niches to their modern descendants. Predators in streams and rivers hunting smaller invertebrates. Fossil evidence from Carboniferous freshwater deposits occasionally preserves stonefly nymphs alongside their prey, providing snapshots of feeding interactions. These nymphs possess strong jaws and grasping legs adapted for ambush hunting, waiting among rocks and vegetation for passing victims. The high oxygen levels in Carboniferous waters, equilibrated with the hyperoxic atmosphere, allowed these larvae to grow larger than modern stonefly nymphs. Adult stoneflies were weak flyers then as now, preferring to walk or run across surfaces rather than fly long distances. Their legs show adaptations for gripping rough bark and rocky surfaces, allowing them to navigate complex three-dimensional environments in forests and along stream banks. The males likely used substrate vibrations for communication, drumming their abdomens against wood or stone to attract females, a behavior modern stoneflies still employ. The specific rhythms and frequencies would have been species-specific, preventing cross-species confusion in areas where multiple stonefly species coexisted. Mating in stoneflies involves males locating drumming females and approaching for copulation. Carboniferous species probably followed similar patterns, with males competing for access to receptive females through drumming contests and direct confrontation. After mating, females would have sought suitable oviposition sites in streams or rivers, depositing eggs that sank to the bottom and hatched into predatory nymphs. The life cycle would have spanned years, with nymphs growing through numerous molts before emerging as adults for brief reproductive periods. The dietary preferences of adult stoneflies remain somewhat mysterious. Modern species rarely feed as adults, focusing entirely on reproduction during their brief wing stage. Carboniferous stoneflies may have followed similar patterns, though their mouthparts show slight differences, suggesting they might have consumed lichens or other simple plant material. These feeding behaviours would have been minimal compared to the intensive feeding during larval stages. Fossilised stonefly swarms occasionally appear in lake deposits, showing that some species engaged in synchronised emergencies similar to mayflies but on smaller scales. These emergence events would have attracted predators and created temporary feeding opportunities for birds. Though, of course, birds hadn't yet evolved, so aerial predators like Meganeuropsis would have capitalised instead. The timing of emergencies likely coordinated with environmental cues like temperature and photoperiod. ensuring adults emerged when conditions favoured survival and reproduction. Stoneflies serve as excellent indicators of water quality both today and in the fossil record. Their presence indicates well-oxygenated flowing water, while their absence suggests pollution or stagnant conditions. The distribution of Carboniferous stonefly fossils maps the locations of healthy stream systems, revealing where ancient rivers flowed through forests and provided the precise habitat conditions these insects required. The evolutionary conservatism of stoneflies, maintaining the same basic body plan for over 300 million years, demonstrates that some successful designs need little modification. While other insect groups experimented with diverse forms, stoneflies found an effective ecological strategy and refined it incrementally rather than revolutionizing their anatomy. This consistency makes them valuable for understanding ancestral insect biology and tracking how environments changed over geological time. The primitive beetles. Beetles represent the most diverse insect order today, but their Carboniferous ancestors were relatively few and simple compared to later radiations. Early beetles, like Moravicoleus, lacked many features we associate with modern beetles: sophisticated flight mechanisms, specialized mouthparts for diverse diets, and the hardened elytra wing covers that characterize most modern species. Instead, they represented evolutionary experiments in a new insect body plan that would eventually dominate terrestrial ecosystems. The wing structure of primitive beetles shows they were in transition between soft-winged ancestors and the armored forms that would later evolve. The forewings were partially hardened, providing some protection for the hindwings, but not the complete coverage seen in modern beetles. This intermediate stage reveals how wing covers evolved gradually, with each incremental improvement in protection conferring selective advantages. The hind wings remained membranous and functional for flight, though fossil evidence suggests these early beetles were probably weak flyers compared to dragonflies or even mayflies. The jaws of Carboniferous beetles indicate generalized feeding habits. Unlike the highly specialized mandibles of modern beetles adapted for cutting leaves, boring through wood, or predation, early beetle jaws were simple structures suitable for consuming soft plant material and possibly scavenging. The lack of specialization suggests these beetles were opportunistically fed on whatever was available: decaying vegetation, fungal growths, possibly soft-bodied arthropods, rather than committing to specific food sources. Body armor in primitive beetles was less developed than in later forms. The exoskeleton provided basic protection, but lacked the thick, heavily sclerotized plates of modern beetles. This made them more vulnerable to predators, but also lighter and potentially more mobile. The trade-off between protection and mobility shapes arthropod evolution broadly, and early beetles were still exploring different points along this continuum. Fossil preservation of beetles is often excellent due to their hardy exoskeletons. Specimens from Carboniferous deposits show fine details including joint articulations, muscle attachment points, and even impressions of soft tissues. These fossils allow researchers to reconstruct beetle anatomy with precision, revealing how internal structures supported their lifestyle. The digestive system, though rarely fossilized, can sometimes be inferred from gut contents showing what these insects consumed. Habitat preferences of early beetles centered on forest floor environments where dead plant material accumulated. The rotting scaled tree trunks and leaf litter provided food and shelter, allowing beetle populations to thrive alongside millipedes, centipedes, and other detritivores. Competition for resources would have driven some specialization, with different beetle species focusing on different decay stages or plant types. Larval stages in primitive beetles probably resembled modern beetle larvae, soft-bodied grubs that lived in soil or rotting wood. These larvae would have fed intensively, accumulating resources for the energy-demanding transformation to adult form. The pupation stage, a period of metamorphosis, would have occurred in protected locations, with the larval body completely reorganizing into adult structures. This complete metamorphosis distinguishes beetles and other holometabolous insects from more primitive groups that develop gradually through incremental molts. The evolutionary success of beetles stems partly from their ability to exploit diverse food sources and habitats. Even in the Carboniferous, early beetles were beginning to explore different ecological niches. Some species show adaptations for living under bark, others for burrowing in soil, and still others for aquatic environments. This niche diversification laid the groundwork for the explosive beetle radiation that would occur in later geological periods. Chemical defenses may have already evolved in Carboniferous beetles. Modern beetles employ an astonishing array of toxic compounds, irritating secretions, and deterrent chemicals to ward off predators. While direct evidence of chemical defenses in fossil beetles is impossible, the presence of defensive glands in some specimens suggests these strategies were already developing. The arms race between beetles and their predators, which includes everything from other arthropods to amphibians to early reptiles, would have favored the evolution of any advantage, including chemical warfare. Stem tetrapods, early amphibian predators. The Carboniferous wasn't exclusively ruled by arthropods. Early amphibians, stem tetrapods that bridge the evolutionary gap between fish and true land vertebrates, prowled the same environments as giant insects and spiders. These primitive four-legged animals had evolved from lobe-finned fish during the preceding Devonian period, and by Carboniferous times had diversified into numerous forms, many of which preyed upon the abundant arthropod fauna. Creatures like Crassigyrinus exemplify these early amphibian predators. Growing to nearly 2 meters in length, Crassigyrinus possessed a massive head, powerful jaws lined with sharp teeth, and a relatively slender body with weak, small limbs. Its anatomy suggests it was primarily aquatic, hunting in Carboniferous swamps and rivers where it pursued fish and large aquatic arthropods. The weakness of its limbs indicates limited terrestrial capability. Crassigyrinus could probably pull itself onto mudflats, but wasn't adapted for sustained walking on land. The skull structure of early amphibians reveals specializations for aquatic hunting. The bones forming the roof of the mouth contain numerous small teeth in addition to larger marginal teeth, creating a trap that prevented prey from escaping once caught. The jaw joint allowed wide gape angles, enabling these predators to engulf relatively large prey whole. Fossilized coprolites associated with early amphibians occasionally contain recognizable arthropod fragments, confirming they fed on the available insect and arachnid fauna. Other stem tetrapods adapted for more terrestrial lifestyles. Proterogyrinus achieved lengths exceeding 2 meters and possessed stronger limbs capable of supporting its weight on land. The proportion of its skeleton suggests it spent time both in water and on land, hunting along shorelines where aquatic and terrestrial environments intersected. Its diet likely included fish, smaller amphibians, and terrestrial arthropods, including scorpions, millipedes, and possibly even juvenile Arthropleura. The respiratory systems of these early amphibians were still primitive compared to modern tetrapods. While they possessed lungs capable of breathing air, they also retained functional gills or relied on cutaneous respiration, gas exchange through moist skin. This limited them to humid environments where skin breathing remained effective. The hyperoxic Carboniferous atmosphere would have made respiratory adaptations less critical than they would be in later, lower oxygen environments, allowing these animals to function with less efficient breathing systems. Reproduction in stem tetrapods remained tied to water. Like modern amphibians, they laid eggs in aquatic environments where larvae developed with external gills before metamorphosing into adults. The larval stage probably lasted months to years, depending on species and environmental conditions. During this time, larvae were vulnerable to predation from both aquatic arthropods like eurypterids and adult amphibians, including cannibalistic members of their own species. The skin of early amphibians likely lacked the impermeable scales of later reptiles, remaining permeable to water and requiring the animals to stay near humid environments to prevent desiccation. This constraint limited their distribution to areas with permanent or seasonal water bodies. Carboniferous swamp forests provided ideal habitats, but drier upland regions remained inaccessible to these moisture-dependent predators. Competition and predation dynamics between arthropods and early amphibians shaped both groups. Young amphibians faced threats from large scorpions and aquatic eurypterids, while adult amphibians could prey upon all but the largest arthropods. This created complex food web interactions where the same species occupied different trophic levels depending on life stage. A larval Proterogyrinus might be prey for Pulmonoscorpius, while an adult could hunt the same scorpion species. The evolution of tetrapods during the Carboniferous set the stage for the eventual rise of reptiles and mammals. The innovations in limb structure, jaw mechanics, and sensory systems that appeared in stem tetrapods would be refined and elaborated in their descendants. But during the Carboniferous itself, these early amphibians remained secondary to arthropods in terms of diversity and ecological dominance, interesting predators in a world still ruled by insects and their relatives. The breathing challenge. Understanding why Carboniferous arthropods grew so large requires examining the fundamental physics of how they obtain oxygen. The tracheal system used by insects and other terrestrial arthropods is elegantly simple, but comes with strict size limitations. Air enters through spiracles, holes along the body surface, and travels through progressively narrower tubes that penetrate deep into tissues. Oxygen diffuses directly from these tubes to cells, bypassing the need for circulatory systems to transport respiratory gases. This diffusion-based system works beautifully for small animals. At small scales, diffusion occurs rapidly enough to meet metabolic demands even during intense activity, like flight or running. But physics imposes harsh limits as body size increases. The rate of diffusion depends on the concentration gradient and the distance gases must travel. As arthropods grow larger, the interior tissues become farther from spiracles, requiring oxygen to diffuse through longer tracheal tubes. The concentration gradient remains fixed by atmospheric oxygen levels, so the only variable that can compensate for increased distance is time, meaning larger arthropods must either slow their metabolism or find ways to increase oxygen availability. The Carboniferous atmosphere solved this problem by dramatically increasing oxygen concentration. With atmospheric oxygen at 35% instead of today's 21%, the concentration gradient driving diffusion into tissues steepened considerably. This meant oxygen could diffuse farther into the body before becoming depleted, effectively extending the maximum distance from the spiracle to deep tissue that could still receive adequate oxygen supply. Mathematical models of gas diffusion show that a 35% oxygen atmosphere allows arthropods to reach roughly twice the linear dimensions possible in modern atmospheres while maintaining the same oxygen delivery rate to tissues. But higher oxygen levels create their own problems. Oxygen is chemically reactive and can damage cells through oxidative stress. Modern organisms possess extensive antioxidant systems to protect against oxygen damage, but these defenses come at metabolic cost. Carboniferous arthropods would have required enhanced antioxidant capabilities to survive in hyperoxic conditions. Evidence for this comes from biochemical analysis of arthropod evolution, showing that genes encoding antioxidant enzymes underwent selection during periods of high atmospheric oxygen. Temperature also affects respiratory efficiency. Higher temperatures increase metabolic rates, demanding more oxygen delivery, but they also speed up diffusion rates. Carboniferous climates were generally warm and stable, creating conditions where respiratory systems operated near optimal efficiency. The combination of high oxygen, warm temperatures, and stable conditions created a sweet spot for arthropod gigantism. The evolution of book lungs in arachnids like scorpions represents an alternative solution to the scaling problem. Book lungs consist of stacked plates with thin membranes where gas exchange occurs. Blood flows through these plates, allowing oxygen to diffuse into the circulatory system rather than directly to tissues. This system is more efficient than trachea for larger body sizes because it uses active circulation to transport oxygen rather than relying solely on passive diffusion. The combination of book lungs and high atmospheric oxygen allowed Pulmonoscorpius to achieve sizes impossible for purely tracheal arthropods even in hyperoxic conditions. Modern experiments manipulating atmospheric oxygen levels confirm these principles. When contemporary insects are raised in high oxygen environments, they grow larger than controls in normal air, though the effect is modest compared to the size increases seen in the fossil record. This demonstrates that oxygen availability does influence achievable body size, though evolutionary adaptation is also necessary to fully exploit high oxygen conditions. Carboniferous arthropods had millions of years to optimize their body plans for gigantism, explaining why they achieved sizes far beyond what short-term laboratory experiments produce. The end of atmospheric hyperoxia doomed giant arthropods. As oxygen levels declined through the Permian period, the respiratory systems of giants became inadequate. Unable to modify their fundamental physiology quickly enough, large arthropod populations crashed. Smaller species, with their shorter diffusion distances and lower oxygen demands, weathered the transition more successfully. This created a size bottleneck that arthropods have never escaped. Modern atmospheric chemistry simply cannot support insect giants. Fire and forest dynamics. The hyperoxic Carboniferous atmosphere had consequences beyond enabling arthropod gigantism. High oxygen concentrations made organic matter significantly more flammable, fundamentally altering forest ecology through frequent, intense wildfires. Modern forests burn when temperatures, drought, and ignition sources align, but Carboniferous forests could ignite even in relatively wet conditions due to the oxygen-rich atmosphere. This created a fire regime unlike anything in modern ecosystems. Evidence for widespread Carboniferous fires comes from charcoal layers in coal deposits. Fossilized charcoal, termed fusaan by geologists, appears throughout Carboniferous rock sequences, often in association with well-preserved plant fossils. The abundance and distribution of fusaan indicate that fire was a regular ecological force rather than a rare catastrophe. Chemical analysis of fusaan shows it formed at high temperatures consistent with crown fires, the most intense type of wildfire where flames reach into tree canopies. Ignition sources in the Carboniferous were primarily lightning strikes. Volcanic activity may have contributed in some regions, but most fires probably started when electrical storms struck forests. Once ignited, fires would have spread rapidly through areas with accumulated dead plant material. The oxygen-rich atmosphere meant that even green, living vegetation could burn if fires were intense enough, allowing flames to spread through areas that would be fire-resistant in modern conditions. Plant adaptations to fire-shaped Carboniferous forest composition. Some species evolved thick bark that protected their growing tissue from heat damage. Others produced specialized structures that sprouted quickly after fires, allowing rapid recolonization of burned areas. The scale trees, despite their success in swamp environments, were vulnerable to fire due to their relatively thin bark and shallow roots. Large fires would have killed vast swaths of scale trees, opening areas for colonization by more fire-resistant species. Fire created heterogeneous landscapes with patches at different successional stages. Recently burned areas would have been colonized by pioneering species, probably including certain ferns and seed ferns that produced abundant wind-blown spores. These pioneer communities provided different resources than mature forests, supporting distinct arthropod assemblages. As forests matured, the vegetation structure became more complex and arthropod diversity increased accordingly. Eventually, fuel accumulation and lightning strikes would trigger new fires, resetting the cycle. The impact of fire on arthropod populations varied by species. Mobile animals like flying insects could escape advancing flames, though widespread fires might trap even strong flyers. Ground-dwelling arthropods faced higher mortality, particularly species like Arthropleura that moved slowly. However, the rapid regrowth of vegetation after fires would have created resource pulses that supported explosive population growth in surviving insects and those that colonized from unburned areas. Fire frequency remains difficult to determine precisely from fossil evidence, but the abundance of charcoal suggests fires occurred on timescales of decades rather than centuries. Some areas probably burned multiple times per century, preventing the development of truly old-growth forests. This frequent disturbance would have favoured R-selected species, organisms that reproduce quickly and colonize new habitats rapidly, over K-selected species adapted to stable, mature environments. The interaction between fire and coal formation created feedback loops. Fire converted some plant material to charcoal, making it more resistant to decomposition and likely to contribute to coal deposits. But fire also released carbon as carbon dioxide, partially counteracting the carbon burial that drove oxygen levels upward. The balance between these processes influenced long-term atmospheric composition and climate. Human understanding of Carboniferous fire ecology has important implications for interpreting ancient environments. The presence or absence of fusain in fossil deposits helps paleontologists reconstruct fire frequency and intensity. Patterns of charcoal distribution reveal information about wind directions, topography, and vegetation types. This makes fire-related fossils valuable proxies for environmental conditions that are otherwise difficult to reconstruct. The microscopic world. While giant insects capture imagination, the Carboniferous microscopic realm was equally diverse and arguably more important ecologically. Bacteria, fungi, and protists performed essential ecosystem functions: decomposition, nutrient cycling, symbiotic relationships that supported the visible megafauna. Understanding these microorganisms provides crucial context for why Carboniferous forests function so differently from modern ecosystems. Fossilized bacteria from Carboniferous deposits show morphologies similar to modern forms, indicating that basic bacterial designs were already established. Cyanobacteria, photosynthetic bacteria that contributed to oxygen production, thrived in aquatic environments, forming mats and stromatolites in shallow waters. Heterotrophic bacteria that decomposed organic matter were also present, though their capabilities differed from modern bacterial communities. The key difference was in lignin decomposition. Modern forests contain numerous bacteria and fungi capable of breaking down lignin, the complex polymer that gives wood its strength. But during the Carboniferous, these lignin-degrading organisms were either absent or inefficient. This meant that dead-scale trees and other woody plants accumulated rather than being rapidly recycled. The persistence of lignin-rich tissues in oxygen-poor swamp waters led directly to coal formation, fossilized forests representing millions of years of undecomposed plant material. Fungi were present and diverse during the Carboniferous, with fossil evidence showing both saprotropic species that decomposed organic matter and mycorrhizal fungi that formed symbiotic relationships with plant roots. The mycorrhizae help plants access soil nutrients, particularly phosphorus, in exchange for sugars produced through photosynthesis. This mutualism was already well established, indicating these relationships evolved earlier in plant evolutionary history. Some Carboniferous fungi show unusual morphologies suggesting they filled ecological niches that have since disappeared or been occupied by other organisms. Prototaxites, though technically from an earlier period, represent the kind of fungal oddity that populated ancient ecosystems, a structure that grew multiple meters tall with a trunk-like form, unlike any modern fungus. While Prototaxites itself had declined by the Carboniferous, its existence demonstrates that fungal biology explored forms radically different from contemporary species. Protists, single-celled eukaryotes including amoebas, ciliates, and various algae, were abundant in Carboniferous aquatic environments. Fossilized protists appear in fine-grained sediments, though their simple morphologies make species identification challenging. These organisms formed the base of aquatic food webs, converting nutrients and energy from bacteria and algae into biomass that fed aquatic arthropod larvae, worms, and other small animals. The microscopic predators, protists that consume bacteria and other protists, regulated bacterial populations and prevented any single species from dominating. This predation pressure maintained microbial diversity, which in turn supported ecosystem stability. The principles of microbial ecology that govern modern environments were already operating during the Carboniferous, though the specific species involved have long since been replaced by their descendants. Microscopic detritivores broke down fine plant material, converting it into forms accessible to larger animals. The fragmentation of leaf litter by microscopic organisms increased surface area for bacterial and fungal colonization, accelerating decomposition. This process was crucial in nutrient cycling, releasing elements like nitrogen and phosphorus back into soil where plants could access them. The role of microorganisms in insect biology was also significant. Many modern insects harbour symbiotic bacteria or fungi that aid in digestion, produce essential nutrients, or provide protection from pathogens. Evidence from Carboniferous insects suggests these relationships already existed. Chemical traces in fossilized gut contents hint at bacterial communities that may have helped arthropods digest plant material, though direct confirmation is impossible without preserved soft tissues. Disease organisms, pathogenic bacteria, fungi, and protists certainly existed during the Carboniferous. Modern arthropods face numerous microscopic parasites and pathogens, and there's no reason to believe Carboniferous species were immune. Population die-offs from disease would have shaped arthropod populations, potentially preventing any single species from completely dominating ecosystems. The density of organisms in favourable habitats would have facilitated disease transmission, creating natural checks on population growth. The Permian transition. The end of the Carboniferous didn't arrive suddenly, but through gradual environmental changes that accumulated over millions of years. The Permian period that followed brought dramatic climate shifts, declining oxygen levels, and fundamental reorganizations of terrestrial ecosystems. Understanding this transition illuminates why the age of insects ended and why modern arthropods remain comparatively small. Climate change began reshaping Carboniferous landscapes during the late Carboniferous itself. The formation of the supercontinent Pangaea altered global circulation patterns, creating more pronounced seasonality in many regions. Areas that had experienced year-round warmth and humidity began having distinct wet and dry seasons. Some regions became increasingly arid as continental interiors lost access to ocean-moderated climates. These changes stressed swamp forests adapted to constant moisture. The scale trees that dominated Carboniferous forests were particularly vulnerable to drying trends. their shallow root systems couldn't access deep water tables, and their physiology wasn't adapted for water conservation. As climates dried, scaled tree forests retreated toward remaining wetlands, shrinking the habitats that supported Arthropleura and other moisture-dependent arthropods. More drought-tolerant plants like seed ferns and early conifers expanded into areas vacated by scaled trees. Oxygen levels began declining from their Carboniferous peak. The mechanisms driving this decline remain debated, but several factors likely contributed. As scaled tree forests shrank, overall photosynthetic oxygen production decreased. Simultaneously, the evolution of more efficient lignin-decomposing fungi meant that plant material no longer accumulated as readily in oxygen-poor conditions. Dead plants decomposed more completely, releasing carbon dioxide back into the atmosphere and consuming oxygen in the process. This reversed the carbon burial that had driven oxygen levels upward. The consequences for giant arthropods were severe. As oxygen concentrations fell from 35% toward modern levels, the respiratory systems of large species became increasingly inadequate. The diffusion gradients driving oxygen into tissues shallowed, meaning that deep interior tissues received insufficient oxygen. Metabolic rates had to slow, reducing activity levels and competitive ability. Species that had thrived in hyperoxic conditions found themselves suffocating in the changing atmosphere. Body size in arthropods declined progressively through the Permian. Fossilized insects and arachnids show a clear trend towards smaller maximum sizes in late Permian deposits compared to Carboniferous specimens. This wasn't a sudden collapse, but a gradual winnowing as each generation faced slightly less favorable respiratory conditions. Large individuals within species suffered higher mortality, shifting the size distribution of populations downward. Over many generations, average body sizes decreased as selection favored individuals with more efficient respiratory systems relative to their size. The rise of amniotes, reptiles and their relatives, added new competitive and predatory pressures. Early Permian reptiles diversified rapidly, occupying ecological niches that had been largely exclusive to arthropods and amphibians during the Carboniferous. Some reptiles became specialized insect predators, hunting the large insects that were becoming increasingly sluggish as oxygen levels fell. Others became herbivores competing with arthropod detritivores for plant resources. This vertebrate diversification squeezed arthropods from multiple ecological angles. Plant evolution during the Permian transition also affected arthropod communities. The rise of seed plants with more sophisticated defenses, including tougher leaves, chemical deterrents, and better drought resistance, made herbivory more challenging for insects. The relatively undefended Carboniferous flora had allowed arthropod herbivores to thrive with minimal specialization. Permian plants demanded greater specialization, reducing the number of plant species any given insect could exploit. This drove extinction in generalist herbivores while potentially increasing diversity among specialists. The Great Permian extinction at the end of the period, the most severe mass extinction in Earth's history, delivered the final blow to Carboniferous-style ecosystems. Volcanic eruptions in Siberia released massive quantities of carbon dioxide and toxic gases, triggering climate warming, ocean acidification, and widespread oxygen depletion. Both terrestrial and marine ecosystems collapsed, with over 90% of marine species and 70% of terrestrial vertebrates going extinct. Arthropods suffered as well, though their higher diversity and shorter generation times allowed faster recovery compared to vertebrates. The world that emerged from the Permian extinction was fundamentally different from the Carboniferous. Atmospheric oxygen had settled near modern levels. Reptiles dominated terrestrial ecosystems. Flowering plants wouldn't appear for another 100 million years, but the seed plants that survived the extinction diversified into forms more familiar to modern eyes. Insects remained abundant and diverse, but they were small, limited by atmospheric chemistry to sizes that seem modest compared to their Carboniferous ancestors. Legacy and lessons. The age of insects represents more than just a fascinating chapter in Earth's history. It provides crucial insights into how planetary chemistry shapes biological evolution. The Carboniferous demonstrates that life's possibilities extend far beyond what we observe in modern ecosystems. The same evolutionary processes operating today produced radically different outcomes when environmental parameters shifted. The coal deposits formed from Carboniferous forests power modern civilization. The scale trees that died and failed to decompose 300 million years ago became the coal seams that fueled the Industrial Revolution and continue generating electricity today. Every lump of coal represents ancient sunlight captured by photosynthesis and locked away when decomposer organisms couldn't keep pace with plant growth. The irony that we're now releasing that ancient carbon, reversing the process that created the Carboniferous hyperoxic atmosphere, isn't lost on climate scientists. Studying Carboniferous gigantism helps biologists understand the constraints that limit modern organisms. The absence of giant insects today isn't because evolution hasn't figured out how to make them. It's because atmospheric chemistry won't support them. This demonstrates how physical and chemical parameters set boundaries within which evolution operates. Life is creative and adaptive, but it cannot violate basic physics and chemistry. The fossil record of the Carboniferous is exceptionally rich due to the unique preservation conditions in coal swamps. The same anaerobic waterlogged environments that prevented decomposition and led to coal formation also fossilize countless organisms with exquisite detail. Paleontologists can study not just bones and shells, but also soft tissues, behavioral traces, and entire ecosystem snapshots frozen in stone. This makes the Carboniferous one of the best understood ancient time periods. Modern conservation biology can learn from Carboniferous ecosystem dynamics. The interactions between plants, arthropods, fire, and climate created complex feedbacks that shaped evolutionary trajectories. Understanding these relationships helps ecologists predict how current ecosystems might respond to climate change and other anthropogenic pressures. The Carboniferous shows that ecosystems can exist in stable states very different from modern ones. But transitions between states can be rapid and devastating. The engineering principles visible in Carboniferous arthropods inspire modern biomimetic design. The wing structures of Meganeura, optimized for flight in different atmospheric conditions, inform aerospace engineering. The armor systems of giant millipedes and scorpions provide models for protective materials. Even the tracheal breathing systems, though limited at large scales, demonstrate remarkably efficient gas distribution networks that inspire microfluidic device design. Educational value is another legacy of Carboniferous research. The dramatic size differences between ancient and modern arthropods capture public imagination, making insect paleontology accessible to non-specialists. Children fascinated by giant dragonflies often develop interests in science that lead to careers in biology, paleontology, or related fields. Museums worldwide feature Carboniferous displays that communicate scientific concepts through spectacular fossils. The Carboniferous also serves as a cautionary tale about atmospheric composition. The hyperoxic atmosphere that enabled giants also created hazards, increased fire frequency, oxidative stress, and ecosystem instability. The transition back to lower oxygen levels caused mass extinctions. Current human activities are altering atmospheric composition through greenhouse gas emissions, though in different ways than Carboniferous plants did. The lessons of deep time suggest that even small changes in atmospheric chemistry can cascade into ecosystem-level transformations. Future research on the Carboniferous continues discovering new species, refining paleo-atmospheric reconstructions, and testing hypotheses about how ancient ecosystems functioned. Advanced imaging techniques allow scientists to examine fossils in unprecedented detail without destroying them. Geochemical proxies improve estimates of ancient oxygen levels, temperatures, and other environmental parameters. Computer models simulate Carboniferous ecosystems, testing whether our understanding of biological and physical processes can reproduce observed fossil patterns. The age of insects ended over 250 million years ago, but its legacy persists in coal deposits, fossil collections, and the evolutionary history encoded in modern arthropods. Every insect alive today descended from ancestors that weathered the transition from Carboniferous hyperoxia to Permian normalcy. The genetic adaptations that allowed survival through that transition still shape insect physiology. The Carboniferous proves that Earth's history contains wonders matching any science fiction, and that understanding the past illuminates both present and future.